So thank you and welcome to the second in a introduction to fluorescence and microscopy fluorescence microscopy lecture series. So today we'll be going through experimental design, acquisition, and analysis within fluorescence microscopy. So we're in lecture 2 of this series. Okay, so the overview of what we're covering today is we're going to go through some very general advice on sample preparation, how to prepare your samples for performing the imaging. We'll then go through some experimental parameters, and this is focused wholly around confocal imaging in terms of what your settings are and things to consider when you're setting up your confocal to do an experiment. We'll then go through what you do with your image once you've actually generated it. So a little thing around image ethics, what you can do, what you shouldn't do in terms of post-acquisition on your image. And then finally, I'll give a very, again, general overview of kind of image analysis workflow and considerations that you have to take into account when you're going through image analysis. So let's start up with sample preparation. So I wanted to start with a few kind of general rules in terms of how best to go about sample preparation. And in general, for fluorescence microscopy, the best imaging is going to occur near the cover slip surface. So as we mentioned on Tuesday, the objective you're using is normally expecting a cover slip with about 170 micron thickness. That's a number 1.5 cover slip. So you'll get the best image quality if you're imaging through a cover slip with the sample mounted directly on the other side of the cover slip. If your sample is mounted on the slide or you're trying to image through a slide, that glass is obviously thicker and therefore you'll get lower quality images. Next thing, and again, we touched on this on Tuesday, was to choose the best fluorophore for your experiment. After that, we can try and match those fluorophores to a microscope setup. So obviously, we'll cover this in the confocal section in terms of, matching the laser excitation to the fluorophores that you're using in your experiment. The next thing, which I always preach often to people that won't listen, is to titrate your concentration. So if you're doing transfections, then don't just go in with the concentration that was used or that it says in the protocol from the manufacturer. Actually go through and do a bit of work and actually optimize each of your experimental steps. So things like the DNA for transfections, things like your fixative concentrations and the ratios of those fixatives, things like your antibodies, or if you're using dyes or stains, the concentrations of those. And something that's really, really important, and we touched this on Tuesday, is that we're still doing an experimental project, even though we're generating essentially images at the end. So we still need positive and negative controls within our experiments. And you wouldn't run a Western blot without a positive or a negative control. So why would you run a microscopy experiment without them? Next thing is the fixatives. So try and use as gentle a fixative to preserve your sample as you can. You can introduce a lot of artifacts into your samples by over-fixing or using the wrong fix for your sample. And then we suggest that you try and image your samples as closely to preparation as possible and trying to make that consistent across multiple experiments as well. The final thing on the general rules here are to use the correct sample mounting for your microscope. So that's what you're putting your sample in when you're mounting the cover slip on. And that will greatly depend on your objective that you're using. So here for an oil lens, we've got a sample which is mounted in something with a refractive index of 1.33. That's about water. You can see the image quality isn't as good as if you've matched the refractive index with something more like a glycerol-based mounting media. Okay, so the reason I give general sample prep rules in this talk is because there are a million different ways that you can prepare samples. So it's very, very difficult to give, one size fits all rules. This is your kind of decision tree in terms of what you're doing for sample prep. So I would encourage you as much as possible to go down the live route. There's nothing better than acquiring data from a sample which is still living and still going about it semi-normal life if it's on a dish. If you are going down the live route, then mostly you'll be looking at using fluorescent proteins or dyes. If you do decide to fix your samples, then that obviously opens you up to antibody labeling as well as using fluorescent proteins and dyes as well. So we'll go through a few of the different options and things to be aware of in terms of live versus fixed sample prep over the next few slides. Okay, so in terms of live cell imaging, the main thing that you want to do is to keep your cells alive and happy for the duration of your imaging experiment. There's no point trying to image cell division if your cells are dying halfway through your experiment. So first thing is to keep the temperature at a level that the cells are expecting. So normally if we're culturing eukaryotic cells, we're doing that at 37 degrees. You'll often see the microscope with one of these chambers around the outside, which allows you to heat that environment and keep the cells nice and warm so that they can continue their normal existence. Very often you'll find microscopes have little environmental chambers which will go just around your sample. This is very often to control both the gaseous environment. So your cell incubator normally has 5% CO2 in there. That's to maintain the pH balance in your sample. So we want to do that on the microscope as well. So you've got a gas line coming in here, pumping CO2. Very often that gas is going through some kind of bubbler system like shown here. And that's to just make sure that the humidity which is going onto the sample is fairly high. So we reduce the effect of any evaporation on your samples as well. Again, you don't want to get to the bottom of the end of your experiment, say, well, I've still got about a third left of the media that I put on there at the start. That's not a good way to run the experiment. And then the final thing is just to make sure that, the cells are going to be happy for the duration of your experiment. So you've got enough nutrients in there to keep them going for that duration. Okay, in terms of microscopy setup, so choosing the right microscope to do your experiments. So there's a few options here that we have within our platform. So the spinning disc I already mentioned on Tuesday is very, very gentle on samples. So ideally set up for live cell imaging. If you have a confocal microscope with what's called a resonance scanner, which can scan across your sample extremely fast, that again is very, very, very gentle on the imaging. And we also have some of the AeriScan systems, which allow you to also scan very, very fast as well in multiplex mode. I'd say the system I probably should have added in here was the lattice light sheet that we briefly discussed on Tuesday. And again, that is very, very gentle on your samples. Next thing is to select the correct fluorophore. So you want them as bright as possible, so you can keep your excitation levels as low as possible. And you probably want to focus more on the longer excitation wavelengths, so green, red, far red. Normally, I would suggest not putting things like DAPI or Hurst into your live cell experiments because you're exciting with an ultraviolet light. And you know how humans don't like UV, so cells also do not like ultraviolet light as well. That goes on to the reduced phototoxicity, so longer wavelengths if we can, and also try and make sure that those excitation levels are used at the minimum that we can actually get away with for the experiment. Final thing is how we're mounting the samples. So again, we're expecting that cover slip thickness, either in glass or in a plastic. You can get chambers like these that you can assemble together. So you have your cover slip here with your cells on. You assemble this and put the media in. People will 3D print their own of these now. Otherwise, there are commercially available ones. Otherwise, you can use imaging dishes. These are examples from a company called iBiddy, or there are fluoro dishes are available as well. the main thing is that you have that cover slip thickness base to get the best image quality that you can. All right, so that's as deep as I want to get on the live cell imaging. We'll now go into sample fixation, and there's again a number of different avenues we can go down. So we'll go through each one in turn through the next slides. I guess the main kickoff point here is how we're fixing the samples and what fixing in quotation marks does. So when we're fixing our samples, we're creating a chemical cross-link between proteins within the samples. This stops the samples from degrading, kind of captures it in a moment, I guess, that we can then use to stain later on. Some of the common fixes which are used are things like glutaraldehyde, which is a very strong fixative, but doesn't always provides good morphology, but poor stainings. Another common one is paraformaldehyde, which is a weak fixer, but provides poor morphology, but good staining. So normally you'll find that a lot of protocols will use a combination of glutaraldehyde and paraformaldehyde together to get a kind of halfway house between those two fixatives. Some others that can be used are things like acetomethanol. These very often are very harsh fixatives and cause a lot of artifacts. There is a relatively new one out there called glyoxyl, which is kind of a nice middle ground between glutaraldehyde and paraformaldehyde. Again, this is worth spending a bit of time at the start of your project to optimize the fixation protocol for what you're doing. And depending on cell type, tissue type, structures that you're looking at, what fixatives you use can change. Okay, so if we look at a kind of cell culture experiment where we're fixing cells, say on a cover slip, to perform an immunofluorescence assay. So in this case, grow or attach our cells to the cover slip. So if your cells are adherent and will attach to the cover slip, that's great. Otherwise, you can use things like polyl lysine, or there's a commercially available thing called CellTac, where you can attach your cells onto the cover slip. Again, you're going to get the best imaging possible at that cover slip interface. You then want to fix your cells, and again, I mentioned there are multiple different ways. It's worth spending time titrating concentrations and conditions to try and work this up to get the best possible results. The next thing we want to do, if we're going to do an immunofluorescence assay, we need to get those antibodies into the cells. So we're going to permeabilize the cells using something like Triton X100. And again, play around with concentrations and also different permeabilizing agents. You'll find, for example, if you're trying to get into different organelles, that different permeabilizing agents work differently on different organelles within the cells. Next thing before we do the antibody label is we want to go through a block step. So we're going to use something like bovine serum albumin, which is basically just a mixture of high concentration proteins. And this will basically randomly bind across your sample and will help reduce any non-specific antibody binding. We then go through the antibody labeling, and I'll go through that a little bit more in depth in a few slides' time. And then finally, we mount the sample for imaging. If we're looking more at tissue sections, then there are a couple of options in terms of where we go. We can either look at paraffin-embedded samples, or we can cryopreserve the samples and look at them that way. So we'll go through. I've got a slide for paraffin and a slide for cryo. So you can isolate the tissue so this is for the paraffin embedded. To maintain the morphology and the structural antigenicity of the sample, we're going to go through chemical fixation as before. So something like formalin or paraformaldehyde or something like that. We then embed the sample into the paraffin, and to do that we need to dehydrate the sample first before we embed it into the paraffin. And the paraffin's giving us something that we can actually cut against when we come to section. So in this case, we're going to cut very, very thin sections, something in the order of 5 to 10 microns, depending on what you want to do. And we're using those a microtome to do that. And I should say all of the histology related things Chris Freelance from our histology facility will cover in lectures 5 and 6 of this series. Once we've sectioned, we'll rehydrate the sample, and then we can go through antigen recovery. So either a heat or enzymatic recovery of the antigens. These are the bits of protein that we're going to detect with the antibodies. And then we go through again the block and label that we talked about at the previous slide as well. If you have any questions around histology, then this website here will take you through our histology facility. The other option I mentioned is to cryopreserve, so freezing our samples. So in this case, again, we're going to go and isolate the tissue, go through a chemical fixation step. But in this case, we infiltrate in sucrose, which gives us some form of cryoprotection. We then embed in something called OCT, optimal cutting temperature. And once we're in the OCT, we can actually cryogenically freeze the samples using something like isopentane, liquid nitrogen, or dry ice. We'll then section using a specialist microtome called a cryostat, which basically everything is kept at very low temperature. And again, in that kind of 5 to 15 micron section thickness. We can air dry these onto a slide, and then we can store those if we need to for long-term up to a year at minus 80 degrees C. Once you're ready to do the staining, you just rehydrate, again, do the blocking step, and then go through the antibody labeling. Okay, so for each of the 3 examples that I've just shown you, so the fixed cells, the paraffin embedded, and the cryopreserved, we need to go back in very often in this case and label proteins that we're interested in with specific antibodies. So in this example, I'm going to go through the indirect immunofluorescence assay or immunohistochemistry assay, where we're using 2 separate antibodies to create our fluorescence signal. So our primary antibody is going to be the one that is detecting our protein of interest. And we need to be aware of which animal the primary antibody was raised in. So I've got an example down here. If our cells are expressing GFP, but we want to antibody label it, we could use a primary antibody, which is anti-GFP, which has been raised in a mouse. Okay, so we go through that. Again, we have to titrate the concentrations to work out what's going to work best. We'll then wash to get rid of any non-specific binding before we come back in with the secondary antibody label. So you remember from the indirect immunofluorescence, the primary antibody is binding our protein of interest. The secondary binds the primary antibody, and it's the secondary that has the fluorophore attached onto it. Again, we would wash that thoroughly, and then optionally, you can fix the samples again just to cross-link those antibodies together, go through another wash step before you mount the sample. So our example down the bottom here, our first antigen is a green fluorescent protein. Our primary antibody is a mouse anti-GFP, so it's recognizing the GFP molecule, and it was raised in mouse. Our secondary antibody is a goat anti-mouse, so it's recognizing the anti-mouse antibody, but it was raised in goat itself, and it's been conjugated with an alexafluorophore-88 fluorophore. In the other example, we have actin. That's an anti-actin primary raised in rabbits. And in this case, we've got the anti-rabbit secondary, which has been raised in sheep, and that has an alexafluor-568. So these 2 stains could be used in combination to give you a double stain on your sample. Okay, let's just go through the experimental parameters. So that's all I really wanted to cover on sample prep. It is very general and high level, But again, if you ever have any questions, then either reach out to your microscopy core or your histology core to get more advice on your specific case. Okay, the experimental parameters, as I mentioned at the start, are really about trying to teach you guys in terms of what's really important in setting up your experiments. So here at the facility, we spend a lot of time training people to use the microscopes to generate these nice, pretty pictures. And the thing that sits in between is these really, really complex software interfaces. And that's what we spend a lot of time training users to understand what those settings should be. So this is an overview of what we need to take into consideration when we're setting up the confocal parameters. So each software has its own different user interface. So we're going to be fairly vendor agnostic here and just go through the kind of key parameters which are in all of the softwares. Okay, and these are our laser settings, our scanning sequence, the objective lens and numerical aperture, the pinhole settings that we talked about last time, frame size, so how many pixels our image is made up of, our gain and our offset, which are detector settings, our bit depth settings, our scan speed, and then finally something called averaging. Okay, so let's start with laser. So each instrument will come with slightly different setups in terms of lasers which are available. It's a good idea to check that before you get to the instrument. So here at BOMP, each of our microscopes has a specification sheet which you can check. That's all available on our webpage. If you want to go and have a look and just double check that the system that you want to use has the correct laser. When you're doing that, it's a good idea to check the excitation and emission spectra for your fluorophores. So here we've got the dotted line is our excitation spectra, and the solid line here is our emission spectra. You can do this either before using something like Spectral Viewer. This is available on the Thermo Fisher webpage, or a lot of the systems now, the software actually allows you to bring up the excitation and emission spectra for the fluorescent protein that you're using. When you do come to use it, it's a good idea to check for the autofluorescence for each of the lasers. I would encourage you to do your negative controls when you're first starting up your experiment, just to make sure that the system is set up properly. We saw this slide on Tuesday, but just to remind you that there are a large plethora of laser lines available to you on the confocals. So it's a good idea, as I said, to check and make sure that you've got the laser line that's applicable for your project. And if you don't, it might be worthwhile thinking about changing the fluorophores that you're using for your project. And this is because, if you remember from Tuesday, the blue line here is our excitation spectra. What we want to do is try and match the laser that we're using to the peak of the excitation spectra. That means that we're going to get the most efficient emission coming off that fluorophore. If we use lasers which aren't ideal, you can see that the signal that we're getting back out from emission drops off quite dramatically, and can also cause other problems if you start then exciting other fluorophores in your sample as well. Okay, so let's have a look at this experimental setup. So we've got a 4-channel experiment running here. Our sample has DAPI, FITSI, AlexaFluor 561 and AlexaFluor 633. And we have lasers which are specific for each of those dyes available on the system, which is great. And we also have 4 detectors on this system. So 4 PMT, photomultiplier tube detectors. So in theory, we could run all of these samples at the same time and get an image really, really quickly. The problem with that is that if you do that, you start to see that your DAPI signal starts bleeding through into your FITSI signal. There's a bit of the FITSI signal in your 561 signal, and there's a bit of the 561 signal in your 633 signal. So it's really starting to complicate your analysis. So how can we change and improve that setup? So what most confocals will allow you to do is something called a sequential scan, where essentially we can scan one channel at a time if we wanted to. So in this case, we have the 405 line, which means we're just detecting our exciting and detecting our DAPI channel. We'll have the 488 line, which is just detecting our green channel, in this case, FITC. We have, I think that was 561 laser line, which is just exciting our Alexa Fluor 561. And then 633 is just exciting this 633 Fluor 4. So the nice thing here is we don't have any bleed through because we're catching, or we have minimal bleed through because we're catching individual channels. The downside is it's 4 times slower, right, than the previous example. So normally for fixed sample imaging or live sample, if your samples aren't moving around too fast. So this will give you, in most instances, the cleanest imaging setup. There's a kind of halfway house where you could combine lasers and detectors. So in this case, we've got our FITC and our 633 channels combined on one track or one acquisition. In this case, they're spectrally separated enough that there's not very much bleed through or crosstalk happening here. And similarly, on the second one, we have our DAPI channel and our 633, which again are fairly spectrally separated. This has the benefit. So again, less bleed through and twice as fast than doing 4 individual scans. So very often this is a nice halfway house between the two. Of course, you can always do your simultaneous scans, so all of lasers, all detectors running at the same time, if you can put up with that bleed through that you're getting through. And again, it's worthwhile doing the test experiments to make sure it's not going to affect your analysis. This has the benefit that it's very, very fast, so ideal for live cell imaging. We have some systems available. So I mentioned specialist detectors on Tuesday. We have a few systems around with a 32 spectral array detector, which we can actually linearly unmix and separate out 4 or 5 fluorophores that are imaged at the same time. So that would, if you need to do that, that's probably the approach I would take. Okay, through to the objective choice. So we discussed a little bit when we were going through the structures of the fluorescence microscopes on Tuesday about all the key components of the objective. So there's a couple of things here in terms of what you need out of your experiment. So first thing, obviously, is the magnification. So, what image quality do you really need to get? How close up do you really need to image your samples? One part of that is the numerical aperture, which, as I told you last time, is the readout of the resolution. So the equation for working out confocal resolution is 0.4 times the wavelength divided by the numerical aperture. So you can see it's got nothing to do with magnification. It's all a readout of the NA. So you can have a look here. These are a few examples of different objectives that are available. So either air, which is normally below an NA of 1, or an immersion type, which is normally above an NA of 1 for a number of given wavelengths. As I say, there are equations behind that. So here's an example of the confocal and versus the wide field lateral resolution equations. There's also an equation for the axial resolution, so the Z resolution. You will find that that is about 2 to 3 times worse than in the lateral direction. And this depends on the mounting media that you've used. So there's a refractive index of the mounting media within the equation for calculating the resolution in the axial direction. And again, this table just has an overview of the axial resolution in a glycerol-based mounting medium. Okay, so we mentioned that the higher the NA, higher the resolution, and hopefully this image shows you that. So it's the same field of view imaged at on an air lens at 0.75 NA versus probably oil lens at an NA of 1.4. So again, depending on the resolution that you need, if you just want to count the cell, then probably this is sufficient. If you want to count these individual structures within that cell, then obviously you need to go to a higher numerical aperture. One really key thing is if you are looking to do co-localization analysis, then please use the highest NA objective that you can get your hands on. If you're doing co-loc analysis on a objective with an NA of 0.25, then it's going to look like pretty much everything co-localizes. Nice way to skew your data, but probably not ethically correct. So yeah, best to do as high an NA as possible for any co-localization experiments. Another thing to take into account is the brightness of an objective, so how much light it can capture. And again, there's an equation for that. The two factors that are involved there are the numerical aperture and the magnification. So a couple of rules here. So if you have objectives with the same magnification, then use the lens with the higher numerical aperture to get better resolution. If you've got two lenses with the same NA, then you're better off to select one with a lower magnification to get more light, so get a brighter image. Okay, pinhole, we discussed a little bit in the last lecture. So if you remember, in a wide field system, we are illuminating our entire sample, and we can see fluorescence from not just our current focal plane, but also above and below the focal plane, because we're reading everything out to the camera. On a confocal, what we have is a pinhole or a little iris that we can control the diameter of, which sits in front of the detector. And in this case, this will change our depth of field that we're imaging on. So if we have a large pinhole, we've got a large depth of field, we'll get lots of out of focus light. As we narrow that pinhole down, the image gets sharper, the depth of field is smaller, and then finally we get to a point where we get a nice crisp image. So the question is, what is the setting that we need to have that pinhole at? So this is this image here is what a point of light would look like if we imaged it on the microscope. So it's a kind of top-down view. This central area here is where the majority of the fluorescence actually sits, and then we have these outer rings with decreasing levels of fluorescence. So this is what we call the airy disk, and this central part is what we're using to define the pinhole size. So very often on a confocal, you'll see a button that says 1AU or Area 1. That is setting basically the pinhole size to match the diameter of the airy disk here. That also works in Z as well, so can also change the depth of field, as I was saying. So with a smaller pinhole, we get a thinner, tighter optical section through the sample. Again, there are equations for working out what your optical section thickness is. For those who are into equations, this is quite a complicated one. But again, in most softwares, they will do that calculation for you if you hit the 1AU button there. One thing just to be aware of is that the different fluorophores that you're using will have different sizes of airy disks. So if on the shorter wavelengths, if you set it to 1AU, you'll have a very, very thin optical section. And as you go into the longer wavelengths, so from the blue into green and into red, where your wavelength is increasing, that means that your resolution is also decreasing as well. So your airy disk is getting larger. So if you set each of these to 1AU, you'll find that actually you're sampling different section thicknesses in your sample. So Normally what we suggest if you're doing multispectral imaging or multi-channel imaging, that you match your optical section. So in this case, the pinhole would be slightly more open for the blue channel. The green channel, which we would call the reference channel, which, because it's in the middle of our channels that we're acquiring, is set to 1 AU, and our longer wavelength we set slightly smaller. And that's just to make sure that we're actually sampling in Z the same depth for each of those channels. Not all confocals give you this granularity of being able to set that pinhole, unfortunately. Next thing is frame size. So again, we touched on this. So we're creating a digital image by scanning our lasers across the sample and assigning intensity that comes back into pixels. So if you remember, we were calling it an array of pixels. So it's like an Excel spreadsheet of intensity values. So each pixel has a physical size in the image and also a intensity value associated with it, which is coming from the gray level or the bit depth of the image. So we'll cover this physical size and what the settings should be for that, and we'll also discuss our bit depth settings as well. Okay, so in this case we have an image of a blue duck, and what we're wanting to do is to pixelate that and turn it into a digital image. So we're going to go through a little example here. So what we've done is digitize this into pixels, and if our experimental needs is that we need to count the number of blue ducks in an image, then hopefully you would say at a frame size of 3 by 3 that you can't really say that that's a blue duck or not. If by the time you're sampling 5 by 5, it's probably getting a little bit closer to you being able to say that that is one blue duck. If we go to 10 by 10, then you're really close to being certain that that is probably a blue duck. But by the time you get to 20 by 20, you're pretty sure it's got a beak, it's got an eye, it's got the right shape. You'd be fairly happy to say that that's a blue duck, and you could count that within your image. If you go to 300 by 300 pixels, then you're definitely sure that that's a blue duck, but have you oversampled? You probably could have got away with 20 by 20. So this is a very kind of manual way of looking at it, I guess, but you can normally tell by looking at your images whether or not you have sufficient image quality for what it is that you're trying to extract from that image. Fortunately, there's a bit of mathematics behind this as well. It's called the Nyquist-Shannon sampling rate, or sometimes shortened just to Nyquist. Again, on the confocal, you'll sometimes see buttons that say Nyquist. And this says that in order to convert an analog signal to a digital signal, it needs to be sampled at least 2.3 times of the half cycle, which doesn't really make much sense, but we have this example here. So our blue line here is our analog signal, and in red, we've got our digital sampling of that. So at 0.83 times sampling, you can see we're really not representing the blue line very well with the red line. So it's not a good representation. By the time we get to 1.3, it's better, but again, it's really not that good, especially down in this region here and towards the peaks up here. By the time we get to 2.3, then it's probably close enough. It's not perfect, but it's a fairly good representation. So this is probably enough, whereas these two are slightly undersampled. And by the time we get to 10x sampling, again, it's a really good representation, but is this oversampled, especially when it comes to confocal, because you're scanning across, if you're setting more pixels than you have to, that's going to take a lot longer to scan across that sample. Fortunately, there are calculators available. So this is a very good one on the SVI website, this Nyquist calculator. Again, as I say, on most confocals now, they will have a Nyquist or optimal button that you can push. This table shows the recommended pixel dimensions using the theoretical axial resolution, so your XY resolution, divided by 2.3. So for example, if you're on a 63, 1.4 NA oil objective, and you're imaging in your green channel, then you want to be about 60 to 80 nanometers, I'd say, in pixel to get the best resolution out of that objective that you can. If you go below 60 nanometers, then you're probably oversampling. It's going to take longer to scan across your sample. If, however, you just want to count nuclei, then you obviously don't need a pixel size of 60 nanometers. So you really need to work out what it is that you're trying to extract out of your images before you take the images and put these settings into place. Again, if you're in any doubt, then just reach out to us or your own facility. Next thing is gain and offset. So these are, as I mentioned, detector settings. So the detectors are capturing photons of light and transmitting those into an electric signal. Our gain is amplifying that signal up so that we can actually visualize it. And the offset actually clips off the bottom end, and I'll show you in a couple of slides' time what that does. So the question here is, we have 3 pictures of this mountain. Which one gives you the best representation of the size and what's going on around the mountain? So hopefully not this one, that you can't see the top and you can't see what's going on at the bottom. This one is probably okay. You can definitely see the top, but you're not really that sure what's going on, closer in. This one of the 3 is probably the best because you can see the top and you can also see a little bit more context as to what's happening at the lower level. It'd be great if you could have an image that had, the top of the mountain and then all the way down to sea level, and that sea level is your kind of baseline in terms of the height. So of these 3, this is probably the best of the 3. So we'll show you that, but in a microscopy context. Here we've got an image. So this is a plant sample, which we use for training quite a lot. And it's been acquired, looks in the green channel, at least it has a green lookup table. Here we're using a range indicator, which again, a lot of the softwares have available. And here you can see it's in grayscale. This range indicator actually goes, if there are 0 intensity pixels, they'll come up as blue. It then goes black through gray to white. And if we're ever saturating the detector, then that will appear as a red pixel. Down here, we've got so we've drawn a line across one of these objects here, and we're looking at the line intensity across. So this is obviously one of the brighter bits, and this is in the background. And this is the dynamic range of our detector, so from minimum up to maximum. So in this case, this is the optimal setting. We've got the low range intensities all the way up, and we can still see those high range intensities. In this case, we're probably undersaturated, so we're only using about 50% of the dynamic range of the detector. This is probably okay, but ideally you'd like to use more of the dynamic range if you can. In this case, it might be, you don't have enough gain, or you don't have enough laser excitation on the sample to actually spread out your signal across the dynamic range. What we want to avoid are cases like this. Here you can see our intensity's gone above. We've got lots of red pixels in there, which means we've saturated the detector. So basically, we don't know what's happening above this line. We don't know if the mountain keeps going away up here or if it's actually stopping there. We can't quantify any of the information within these red pixels. In this case, either your gain or your laser is too high, and you need to bring that back to something more like this optimal setting. The other thing you can do is to change the offset. So as I say, the offset basically clips the lower intensity information. I would be very, very careful about using offset on a confocal. We tend to tell our researchers not to use it. I'd rather you capture the lower information lower intensity information, you can always clean that up in post-processing later if you need to, but you can't do that if you've already lost that information. So again, you don't know how far down that mountain goes, to use the analogy. So it's good to try and capture it all within the dynamic range. Next thing is bit depth. So again, a detector setting. So most microscopes or most confocals will allow you to change the bit depth of the image. So this is essentially the number of grayscale values that you have within your image. So for a 2-bit image, you have 4 grayscale values, 4-bit image, 16 grayscale values. So it's 2 to the power of your bit depth. So 2 to the power of 8 gives you 256 grayscale values. This is normally the default setting on systems. We would use that for simple imaging if you're just creating a microscope image for qualitative purposes, just to look at or present in a publication or a presentation. 8-bit is probably sufficient. If you're wanting to do any image quantification, then we'd suggest that you increase up your bit depth to 12 or 16. You can see that greatly increases the number of grayscale values and can really help you in terms of segmenting things out, and also on the quantification side of things again. So for confocals, you can vary this. For camera-based systems, it tends to be more set in place. This is just an example. So here we've got an array of 4x2 pixels. On an 8-bit scale, you can see that these 2 pixels here have essentially the same intensity values. Whereas if we go to 16-bit, we can actually start to separate these values out. Okay, last 2 we'll do kind of together. That's the scan speed and averaging. So I've mentioned all the way through on a confocal, we're scanning the laser across and building up our picture. So we can vary the speeds, certainly on Galvo scanners, we can vary the speeds at which the laser scans across. So the problem you might find is that if you're scanning very, very fast across your sample, you're not collecting very much signal as you're doing that, and therefore your image can become very, very grainy. Certainly if you're not doing anything to clean that up, it can be very noisy image quality. A lot of this might be detector noise because you're down in the kind of low intensity ranges. What we can do in that case is slow down the scan. So we're assigning more intensity into each of the pixels, and that will increase our image quality. One thing you want to find if you go too slow is that obviously you might start to photo bleach your sample, and therefore your intensities decrease off. So again, it's a trade-off in terms of scanning across slow enough to build up a high enough image quality. This is where detector noise comes in. So again, if we're increasing that gain, the amplifications of the signal on the detector, we can also increase the noise component of the detector as well. One way of getting around that is to perform something called averaging, where we take multiple copies of the same image. So in this case, this is an image with no averaging. In this case, we've taken 2 images and averaged each pixel across the image. And by the time we get to averaging of 4, you can see we've really got rid of this detector noise, which we had in the background, and it's giving us a much nicer signal to noise ratio than we would have in either no averaging or the averaging of 2. Again, the downside is that's going to take 4 times slower to acquire your data. So again, you're going to want to put in some averaging possibly, depending on your detector settings, and just make sure it's the minimum amount that you can get away with decent image quality. That is the exact same slide. Okay, so let's keep going. So today's talk won't be as long as Tuesday's talk. So now we've generated our images. We'd like to go through some rules in terms of what you can do to them and what you shouldn't be doing to the images. Okay, so we'll start off with data management. And the first thing we recommend is you always save your images as the raw file extension, and that will be different depending on which of the vendors' microscopes you're using. I would not save your initial image as a TIFF or a JPEG, and this is because saving it in this format not only saves the image, but it also saves the metadata as well. So what lasers you used, what objective, what your laser settings and detectors were, your pixel sizes, that type of thing. These commercially available packages should be able to be opened in a number of softwares. So there's the Bioformats plugin in Fiji, for example, which can help you to open up a large number of these file types. Next thing is, if you are going to export your data out, then we suggest that you save it as a .tiff file. This is a non-compressed version of your original image file. So the file size is large and is mostly useful for data analysis and publication. If you are going to export it out as a JPEG, a GIF, or a PNG file, just be aware that that's a lossy compression, which will make your file size smaller, but can be used for presentation, but not for any data analysis. So at the bottom here, we've got an image which has been exported out as a TIFF and a JPEG, and we've subtracted out the JPEG from the TIFF, and you can see exactly all of the information that's actually been lost between those two exports. So just be aware of what's happening to the data depending on the file extension that you choose. Next thing in terms of data management is to never process or modify the original file. So you always want to keep that raw data file. And if you are going to make any modifications, then we suggest that you make subsequent copies. Right? So you go through whatever process one, you create a file copy, preferably with something in the file name that covers what's happened. If you export out, you've got a copy one. If you do something to that, you've got a copy of what you've done to that. And then, process 4, you've got another copy of that. If anything, this just means that if you have multi-step processes, that if you mess up, you don't have to go right back to the start every time. And the key thing is to record every modification that you make, either in your lab book or, as I say, within the file name itself. Next thing is in data management is to back up the data. So we have a lot of users accessing our instruments at the University of Melbourne, so it's never a good idea to use those instruments as data backup. From time to time, we've had viruses on the systems that completely wipe the data. So it's really important to get that data off the acquisition computer as quickly as possible. As we say, always keep it as the original data file. Once you've got it off, you need to double back it up. And most universities now should provide you with access to a double backup resource. That just means that if one of the locations fails, you've always got a backup copy somewhere else. Certainly at the University of Melbourne, you should have access to research drives that allow you to do that. Again, you need to be doing that for the original image, the modified image, any modification processes, and any quantified results. So it all counts as your data. Next thing is that, as I've mentioned before, although we're creating an image, that is actually scientific data. So it's essentially an Excel spreadsheet of values. So we need to make sure that if we're trying to make any modifications, that we're actually performing a data manipulation in doing that. So the question is, what's ethical and what's not ethical? And that's what we'll cover in the next few slides. There are a number of guidelines around this. It's worth doing a bit of background reading when you come to start publishing. Okay, so I've got a few examples taken from papers. If anyone follows Elizabeth Bick on Twitter, it's entertaining and scary to see what people are doing in their published figures. You don't want to have to retract your paper because you've done the wrong thing when you've put your figures together. So in this case, and we'll focus down on this, is people making modifications to a specific feature within a figure. So in this case, this is our original data. And in this case, they've gone in with Photoshop or something else, and they've removed this band up here. Or they've added in some extra bands down there. And this is, if you know how to use Photoshop, this is terrifyingly easy to do. Obviously, these are unethical things to do to your images. The other thing to bear in mind is if you have gels taken from, or images taken from multiple gels, then they need to be clearly separated out. So that's made really clear. Next thing, we've got an electron microscopy image. The raw image is here on the left-hand side, and what the group's done is to detect some proteins that they're interested in using an antibody. And in electron microscopy, you don't use a fluorophore attached to the antibody, but you use a gold nanoparticle, which absorbs the electrons very strongly and will give you a very dark signal. So this is the raw image. These are the structures they're interested in, these little black blobs here. And what they've done is gone in and increased the contrast just in the structures to make them a little bit more visible. They've also removed out this black blob here for some reason in their image as well. Again, not very ethical. You're making specific changes to specific parts of the image. Hopefully you guys wouldn't ever think of doing that. And in this case, we've got a little bit closer to home. We've got a histology section, and you can see in the example on the left, which is the raw image, we have this fold, which has been removed out in the image on the right. Again, specific feature which has been modified. There are a few things that they could have done. So if it's not interrupting the analysis or the interpretation of the image, then you can leave it in, or you could crop it out. Or if it's really key, you go back to the block and you section it out again, and you try and find an image that doesn't have a fold within it. Next one is a fluorescence microscope image, so something a little bit closer to home. So the image on the left shows a field of view with a number of different cells, all look like they have the same stain. But if we go in and do a simple contrast adjustment, you can actually see that these cells here and this group of cells here seem to have a different background to the rest of the image. So it's almost as if they've been kind of cut and pasted into the image in some way. This obviously, again, is not ethical. A word of warning is that journal editors actually have softwares that look at this type of stuff now. So if you do ever try it, then you will get caught out in terms of doing these types of modifications. In this example, the group has removed the backgrounds here and also cleaned up some of the illumination on the tissue that they're interested in. So in this case, again, unethical, large degree of background alteration. In this one, there's a bit of a shading effect in the bottom right-hand corner. And in the cleaned up image that's been corrected. Ideally, if you notice that, it would be best to sort the illumination out on the system ahead of taking the image. And certainly if it's a bump microscope, we'd ask that you let us know so we can sort that out for you. In this case, this would only be acceptable if there's no change in the actual tissue. So it is just the background that you've changed, but you would have to make a statement in the figure legend to that effect. Okay, so let's look at modifications to the whole image. So we talked before about the dynamic range. So again, this is a line profile taken across this fluorescence image, and we're looking at the intensities here as we go across that line. It looks like we're on an 8-bit scale because it's going up to about 256 grayscale values. And in this case, we can adjust the brightness of the sample, so increasing up the intensity essentially after we've acquired the image. But again, you'd need to make a statement in your figure legend or your materials and methods to say that you've adjusted brightness and contrast for visualization purposes. Obviously, if you go too far and then you're essentially saturating the intensities, you can see that the interpretation of that image and that image are very, very different. So this is not acceptable in that case. In this example, again, they're modifying the whole image. So I quite like this experimental setup. They've got their sample and they're obviously looking at, fluorescence intensity or something. It's fairly similar to their positive control sample, and they've included a negative control in there as well. Again, if we do a contrast adjustment, it seems like our negative control is a completely black image. I would suggest that you would never get a completely black image out of a microscope. You always have some form of noise or something going on in the background. Again, their positive control seems to have a very different background level to their sample, which suggests either they've been acquired differently or they've been post-processed differently as well. So again, probably very unethical. Images that are compared should be either acquired under identical conditions and also post-processed identically as well. So what is the best practice? So what we're trying to do at the microscope is to take the best representative image of what you're seeing through the eyepiece. So if you're seeing a beautiful sample through the eyepiece, you should be able to see a beautiful sample through the confocal. What you don't want to do, and which is very easy to do within the confocal software, is to have a not so good sample through the eyepieces, which, if you increase your laser intensity and your gain, produces a beautiful confocal image at the end. So what we want to make sure is we spend as much time as possible at the start, maximizing our stainings and our sample preparation so we get the best possible resolution. So we just need to be careful how we're setting up the confocal, just to make sure that we're doing it appropriately. Ideally, you want to calibrate to a known standard across your experiments, a positive control, which is going to be the brightest sample. Use, where possible, identical acquisition settings between the samples, identical post-processing between the samples. Retain all of the original files if you need to go back to them, and then also document each step of the process in terms of what you've done to the image as you go through. Okay, last few slides. We're just going to go through a very high-level introduction to image analysis. Probably about 6 weeks ago now, we had a 1-hour lecture, the Introduction to Bioimage Analysis. That's available on our website if you missed it and want to follow along. These are kind of key snapshots out of that presentation. Okay, so image analysis considerations. So it's always worthwhile, before you start your experiments, thinking about what the analysis is that you want to do. So we've got to really consider that at the start in the experimental planning stage, and start that as soon as possible. Don't leave it to the end of your project. So every year we have examples of honors students coming to us who've acquired all their data and they want to do a specific type of analysis, but they haven't acquired it in the right way. So you can't really use it for the purpose they needed it for. And it's because they didn't think of what they were doing before they started the experiment. Okay, so what we want to do is take you from a nice fluorescence image to some form of quantification. But that's actually the last step. So what we want to do first is determine at the start what we need to measure, determine the microscope that we want to use for that, and then go through our sample preparation based on what we're wanting to do. So this is all pre-planning stage of planning out your experiments. Finally, when we come to do the experiments, ooh, my microscope's gone a bit weird. My computer's gone weird. When we actually come to do the experiments, everything's pre-planned. We know what we're doing when it comes to the acquisition, and probably we already know what we're doing in terms of the quantification as well. Okay, so the general workflow is once we have our microscope image, we may want to do some kind of pre-processing to that microscope image. The next step is very often object segmentation, so identifying the objects that we want to quantify before then going on and doing data analysis on them. So we'll go through each of these steps in turn. Data acquisition, so we've covered a lot of this already in the confocal part that we've already discussed today. So sufficient sampling rate, so that's your Nyquist rate, making sure you've got the appropriate number of pixels for what you're trying to resolve within your image. Again, appropriate resolution. I'd also say magnification in there. You need to decide whether or not you're going to be doing quantitative or a qualitative approach. Again, that will inform what you do in terms of your bit depth or your detector settings. Make sure you're avoiding saturation and try and use the whole dynamic range of the detector as much as you can. Next thing in terms of pre-processing, so if we have our fluorescence signal within our object, that gets convolved as it goes through the microscope. So if you think of all the glass that's between your sample and the detector, that's going to cause a few aberrations as it goes through. So there are ways that we can try and clean that up. So things like denoising algorithms are available if our images are really noisy. We can go through and do some background corrections if, again, we've got uneven background, although, as I said, this is probably better to do pre-acquisition. We can do bleaching corrections. So if your experiment's over time and your sample is bleaching, we can try and correct the intensities for that. We can also do that in Z. If you're going through deep samples, your signal gets worse the deeper you go. We can do channel alignment, so sometimes your channels will be slightly off. Certainly if you're doing something like a co-localization analysis, you might want to have a control, some beads there that you can use to align your channels back together again. And then finally, deconvolution, again, as I talked a little bit on Tuesday about that, in terms of reassigning out of focus light and increasing your signal to noise ratio as well, which can often help when you come to do segmentation, which is the next bit. So very often what we're trying to do is to identify regions within our image. So we've got a bunch of green cells here. If we want to look at the area or the intensity of this green region, we can create what we call a threshold, which basically means we're only interested in the intensity values here. So this is our low intensity, the black regions. This is the higher intensity. And we use this to create what we call a binary mask. So we're only going to measure what's within this white region here. Once we have the binary mask, we can do a bunch of things to it. So we can separate out cells based on something called a watershed. We can make the binary mask smaller or larger, depending on how you want to group or separate out objects. We can make individual points for each of the objects, so we can measure distance between them, or we can form skeletons based on the structures of things. The options here are fairly limitless in terms of what you can do. There are now, of course, machine learning algorithms available, which will help you to threshold, especially if you're using things like non-fluorescence images. So things like bright field, phase contrast, color, or electron microscopy images. This uses segmentation, where we're actually teaching the software to identify the different structures. So in this case, we'd go in and identify different cells. This region in red is our cells. The region in green is our background. Here we're identifying 2 different types of tissue. Here we've got a few different types of tissue. And essentially the software will take that information that we've trained it on and help to segment out different parts of the image for us. And there's an example of some of the software is available at the bottom there. Next step, once we've identified the objects that we want to quantify, is actually to read out those measurements. And again, there are thousands of potential measurements that you could make. Very simple ones are things like, count the number of cells within that field of view, look at the intensity within the regions. Has it changed or increased or decreased based on a mutation or something like that? We can look at shape descriptors. What are some quantitative readouts of what this shape is doing in comparison to others? We can look at distance, so how far apart these cells are. We can look at co-localization. If we're doing live cell experiments, we can track the cells, look at their directionality, their speed, that type of thing. Again, thousands of different options in terms of quantification, and it really comes back to the questions that you're trying to ask for your research. This is an overview of the image processing software that's available here at our platform. So a lot of users are using software in the freeware space. So things like ImageJ Fiji, which you can run on your own computer. Cell Profiler, Elastic is the machine learning tool that I was talking about a couple of slides ago. We obviously have licensed software as well from the vendors. Again, a lot of these are either free that you can download and set up on your own computer, or we have licensed versions which are available on our analysis computers. And then we also have the high-end data analysis software. So things like Amaris, Velocity, Amira are used for kind of multi-dimensional data analysis, so visualization and quantification in 3D over time. The SVI is a package which allows you to deconvolve your data, so increase your signal to noise. And Metamorph, again, is more like a licensed version of Fiji, I would say. The nice thing at BOMP is that we run a lot of workshops that cover these software packages. So I mentioned in the last couple of months, we've run our Intro to Bioimage Analysis lectures. We've run our Fiji for beginners, our Fiji for quantification. We've also run an Introduction to Cell Profiler. These other workshops, so the Fiji macro writing, Colloq, Amaris, Huygens, they're planned for later on in the year, but haven't been open for registration yet. If you've missed any of these workshops, then they are available on our website. Or if you're having difficulty tracking it down, just send us an email to this mailing list. Okay, that was a whirlwind tour through sample prep, experimental parameters, image ethics, and image analysis. Hopefully you guys have found the last couple of lectures useful for your research. Certainly helps us in terms of you guys getting up to speed in terms of some of the background in your experiments. If you're ever in need of help, then feel free to reach out to BOMP for any questions that you might have. I'm just going to leave up our workshop schedule for the year. You can have a look through and see if any of these are of interest to you. Most of them will be held face-to-face here in Melbourne. And if you want to be notified about them coming up, then you can either email us to be added onto our mailing list, or you also had the chance to tick that box during registration. Okay, with that, I'm going to stop the recording, and happy to take any questions if you want to put it in